Okay, well, hello, pharmaceutical science students, and welcome to our next lecture in our cell biology topic. So, this is the fifth lecture that you're going to have on cell biology. Altogether, we have 6 lectures, so we're coming towards the end of the topic now. And the final big topic that we're going to be covering is cell membranes. Okay, so this time we're thinking about membrane structure and function and transport. And next time we're going to be thinking about the way cell membranes are involved in interactions between cells. So this is a summary of your syllabus for this part of the topic. So today we're going to be covering cell membrane structure and function, osmotic properties, transport, different sorts of channels, diffusion, all these things. And then next time we're going to be covering cell interactions with their environment and cell-cell interactions. Okay, so membranes in cells. I think that perhaps the picture that you already have of the membrane of a cell, the cell membrane, is something like a bag that holds the contents of the cell in place. Okay, so here we just have a bag, like a plastic bag, like a cavera, with some liquid inside and some things floating around in that liquid. Well, if that's the impression that you have of the cell membrane, you're about to find out that there's an awful lot more to it than that. So if we think of the different membranes in the cells, to begin with, it's not just the membrane around the outside of the cell. There are lots of structures inside the cell that are enclosed in membrane. So the outside of the cell, we have the plasma membrane or the cell membrane. Around the nucleus, we have the nuclear membrane. We have the endoplasmic reticulum, which is made of membrane. We have the Golgi apparatus, which I don't think is illustrated on here, which is made of membrane. We have mitochondria with membrane around them. In plants, we have chloroplasts with membrane around them. We have lysosomes and peroxisomes, all those things that we've learned about, but are separated by membrane from the rest of the cell contents. Okay, so cell membranes are involved in many of the structures that we've seen in the cell, and they're not just passive containers. They play an active and essential role in many of the functions of the cell. So they're not just there holding things in, they're also controlling what passes through the membrane from one side to the other. And the membranes themselves can be important in transporting things around the cell. Okay, so here is a proper diagram of a cell, if you like. And if we look to see how many of these labeled structures have membrane involved with them, we get most of them actually. Okay, so if I start here with the nucleus, it's surrounded by the nuclear envelope. The cell itself, of course, is surrounded by the cell membrane. If it was a plant cell, the chloroplasts would be surrounded by membrane. And here, if we go around, we've got the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi apparatus, a peroxisome, a lysosome, mitochondria, more rough ER, all of them have membrane as an essential part of their structure. So we're going to be looking in detail today at membrane and the functions of membrane. So if you think that what these membrane structures are doing, why they're important in the cell, what the functions of these membranes are. Well, the first important function is compartmentalization, separating the cell into separate compartments, into separate parts, but are able to contain different things and therefore carry out a different function. So, one example that I've already told you about is the lysosome, the recycling center of the cell, responsible for breaking down cell contents when they're worn out. We've seen that inside the lysosome are a lot of digestive enzymes which would break down the cell contents if they're released. In fact, they're important in cell death. When they are released, they are involved in breaking down the whole cell. But in a normal, healthy, functioning cell, the membrane of the lysosome separates those digestive enzymes from the rest of the cell. A second important function we've seen is the nucleus. We've looked at cell division and the way that the genetic material, the chromosomes, are organized and condensed in a way that's possible because they're enclosed inside a nucleus, inside a membrane, inside a separate compartment inside the cell. And we've seen how during cell division, the nuclear membrane breaks apart. It forms tiny vesicles of membrane, which can quickly come back together again to separate the new nuclei. So another important function of the membranes in cells is to contain biochemical activities into a small volume. So this both separates it from the rest of the cell, but it also concentrates that activity. So if you think of a large number of enzymes carrying out a particular activity, if again we think of the digestive enzymes inside the lysosome, they are concentrated inside the lysosome because they're kept in an enclosed space. And therefore, the enzyme and their substrate are close together and allows the enzyme to work very efficiently. Membranes allow selective transport. It allows differences in different compartments. So you'll learn more about how mitochondria work later on. But if we think of a mitochondria to produce energy, it's important to have an electric gradient across a membrane. That's part of the way that the mitochondria is able to carry out its reactions to actually separate different electrical charges on the opposite sides of a cell membrane. If we think of the outside of the cell, the plasma membrane or cell membrane, that is important in receiving stimuli from the environment. So we've talked a little bit about signals being received by the cell when we've thought about something like apoptosis. We talked about hormone signals from outside the cell. Well, of course, if something is outside the cell membrane, it's the membrane of the cell that it first comes into contact with. So that membrane of receptors that are on that membrane are what's important in receiving those signals from outside the cell. And then if we think of the cell as part of a multi-cellular organism, cells interact with other cells. They interact in many different ways. I've given adhesion, sticking together, as an example here. So if we think of a large number of cells in a tissue, that tissue has its structure because the cells interact with each other. They're stuck together, they're adhesive. Okay, so membranes very important in a lot of the things that cells do. Okay, so just a quick recap. I won't go through this. We've seen this slide already, but the membrane around the outside of the cell membrane or plasma membrane, forms a barrier between the outside and the inside of the cell and controls what passes in and out of the cell. So it's not so much like a wall or a thick barrier, it's more like a gate that can open and close and allow things to pass through it. But it's a gate with guards. So the guards make sure that the only things that pass in and out of the cell are the things that are supposed to be passing. And this is what we call selective permeability. Okay, so again, you've seen this before. This is what we've talked about before about the structure of the cell membrane, and we'll be going on to look at this in a lot more detail in a minute. Okay, and the nuclear membrane, just to remind us what we already know, the double membrane with nuclear pores through it. We'll be looking at this in a bit more detail next time, I think. Okay, so that's just to remind us of a nuclear pore. So the next thing I'm going to show you is a video, which it's quite a fun video, quite an amusing video, but it makes some important points about the properties of cell membranes and also gives you a little bit of history about how we discovered cell membranes, how we know about cell membranes. Okay.
Every cell in your body is separated from those around it by its outermost layer, its membrane. A cell membrane must be both sturdy and flexible. Imagine a membrane made of metal, great at keeping the cell's guts inside, but horrible at letting materials flow in and out. But a membrane made of fishnet stocking would go too far in the opposite direction, leaky but easily torn. So the ideal membrane falls somewhere in the middle. Over the past few centuries, we've learned a lot about the way membranes work. The tale starts in the late 1800s, when, according to legend, a German woman named Agnes Pockels was doing dishes. Her observation that not all detergents dissolve grease in the same way piqued her curiosity, so she made careful measurements of the size of soapy films that formed on the surface of a metal tray filled with water. Later, in the 1920s, GE scientists Irving Langmuir and Kathleen Blodgett re-examined the problem with a more elaborate contraption and found that those tiny slicks were in fact a single layer of oil molecules. Each oil molecule has one side that loves water and floats on the surface, and one side that loathes water and protrudes into the air. So what does it have to do with cell membranes? Well, at the turn of the 20th century, chemists Charles Overton and Hans Meyer demonstrated that the cell membrane is composed of substances that, like oil, have a water-loving part and a water-loathing part. We now call these substances lipids. In 1925, two scientists, Evert Gorder and Francois Grendel, pushed our understanding further. They designed an experiment meant to test whether cell membranes are made of only one layer of lipids, a monolayer, or two layers stacked on top of one another, called a bilayer. Gorder and Grendel drew blood from a dog, a sheep, a rabbit, a goat, a guinea pig, and human volunteers. From each of these samples, they extracted all the lipids from all the red blood cells and placed a few drops of this extract on a tray of water. True to form, the lipids, like oil, spread out into a monolayer, whose size Gorder and Grendel could measure. If they compared the surface area of that monolayer to the surface area of the intact red blood cells, they'd be able to tell whether the red blood cell membrane is one or two layers thick. To understand the design of their experiment, imagine looking down at a sandwich. If you measure the surface area of what you see, you'll get the dimensions of a single slice of bread, even though there are two slices, one stacked perfectly atop the other. But if you open the sandwich and place the two slices side by side, you get twice the surface area. The Gorder and Grendel experiment is basically the same idea. The open sandwich is the monolayer formed by the extracted cellular lipids spreading out into a sheet. The closed sandwich is the intact red blood cell membrane. Lo and behold, they observed a 2-to-1 ratio, proving beyond a shadow of a doubt that a cell membrane is a bilayer, which when unstacked yields a monolayer twice its size. So almost 30 years before the double helix structure of DNA was elucidated, a single experiment involving fancy versions of household materials enabled deep insight into the basic architecture of the cell.
Okay, so I hope that that amused you. It's quite a fun video, but I also hope that it reminded you of what you probably already know about the way a membrane is structured. So I'll pause now and remind you to ask questions. I'm quite sad that I haven't had any questions on the cell biology topic on the forum yet. Please do make the use of that. If you're finding this so easy, you don't need to ask any questions. Perhaps you're not thinking deeply enough. Okay, so here is the structure of a cell membrane that we've already seen. So here is the phospholipid bilayer. So two layers of lipid molecules. Lipid is another word for fat. Okay, you'll be doing a lot more about lipids in a few weeks' time in the main biochemistry, the structural biochemistry part of this course. So I'm not going to go into lots of details about lipid structure at the moment. I'm just going to give you a general overview. So two layers of lipid molecules, and we can see those two layers in an electron micrograph. So here is a picture of a cell. Here is the inside of a cell, and here is the outside of the cell. And separating the inside from the outside, we can very clearly see two layers, okay, two lines. This is a technique which uses osmium, a metal, to interact with the outside of a membrane to give it a very black appearance so that we can see it easily with the electron microscope. But if you look, you can see very easily that there is a thick black line, a pale center, and then another thick black line. So this is the lipid bilayer structure of the cell membrane. So here is another picture showing the same sort of thing. So here is an interpretation of what we're seeing. So this is an electron micrograph of a lipid vesicle, a small sphere of lipid. And this is an interpretation of what we're seeing in the way that the molecules are lined up. So the hydrophilic heads of the lipid molecules are on both outside edges of the bilayer, and they are what is able to interact with the osmium and give this dark appearance. And on the inside, there are the hydrophobic fatty tails of the lipid molecules, and they are what gives it this pale light appearance. Okay, so a lipid bilayer, two layers of molecules, and the reason that they line up in this particular structure is because of the way they interact with water. The hydrophilic heads are in contact with water, and the hydrophobic tails are hidden away from water. So each of those lipid molecules has its hydrophilic head, illustrated here as this green sphere, and a hydrophobic tail. So the hydrophilic head is hydrophilic because it's electrically charged and therefore it likes to associate it with water molecules. It's easy to make interactions with the water molecules on both sides of the membrane layer here. On the other hand, the hydrophobic tails are made up of non-polar fatty acids. So as we saw in the video, this is like oil, and you know what it's like. I'm sure that everybody has seen a pool of water with a drop of oil on it, perhaps when you've been cleaning dishes. If you've got a plate which has a lot of oil on it and you put it in the water to clean it, you'll see that that oil floats onto the top of the water. It forms a separate layer. It doesn't dissolve in the water easily. And that's exactly what we have here with our hydrophobic tails. Okay, so a little bit more detail of the structure. There are many different types of phospholipids, so the exact chemical groups that are present in each of these parts of the molecule can be different. So different phospholipids can have different kinds of polar groups, different lengths of fatty acid chain, different degrees of saturation. You'll learn exactly what that means in the main biochemistry in a few weeks' time. But overall, this structure of having a hydrophilic head and a hydrophobic tail is common to all phospholipids. Okay, this is just the same thing drawn out, showing you the actual chemical groups that are involved in the molecule. Again, you'll learn more about that in a few weeks' time. So this particular molecule that's illustrated is called phosphatidylcholine, and it has a phosphate group here and a choline group. So this is its charged head. Okay, so pause and think of any questions, but if it's about phospholipid structure, wait for a few weeks and you'll learn a lot more about that in your biochemistry lectures. Okay, so the next thing I'm going to talk about is the fluid mosaic model of membrane structure. So here we see again our lipid bilayer, but this time we're seeing that it's not just lipids. So here are the lipids illustrated here with blue heads and white tails. Okay, and we've got some other fats involved, some sterols, as these orange structures. But you can also see lots of other things interacting with this lipid bilayer. So here is an integral protein, a protein which is going right through the membrane. Here is a peripheral protein, a protein which is just on one side of the membrane. And here is a peripheral protein which is just on the other side of the membrane. Okay, here's another peripheral one on the inside. So we've got lots of different other things as well as the lipid membrane attached here. So we've got the proteins, the peripheral proteins, and the integral proteins, and we've got other lipids such as glycolipids and sterols. Here's the sterol, as I said, and here's a glycolipid. That's a lipid with a sugar attached to it. Okay, so why is this called fluid mosaic molecule model? Well, when it was first proposed, it was described that the proteins are like icebergs floating in a lipid sea. So I don't know if you've seen a picture of an iceberg, a huge block of ice floating in seawater. But icebergs float with a small amount of ice above the water and a large amount of ice inside the water. Okay. And they're able to move around very easily in the water. So the people who worked out the fluid mosaic model thought that the proteins in the cell membrane were a little bit like that, that they were able to float around, move around, but only in the surface of the water. An iceberg doesn't sink underneath the water because ice floats, nor does it float above the water because icebergs don't do that. Okay, in the same way, the proteins stay in their place within the membrane, but they're able to move around within that membrane. If you don't want to think about icebergs, perhaps you can think of boats floating on a lake. A boat doesn't usually sink underneath the water unless there's something wrong with it, nor does it float above the water, it floats in the water. Okay, and the word mosaic sometimes confuses people. So I thought I'd just explain exactly why the word mosaic is used. So this picture is a mosaic picture made of the coat of arms of Uganda. I'm sure you recognize this. This is actually outside the Bank of Uganda building in Kampala. So it's a mosaic because the picture is made up of tiny little stones or pieces of tiles of different colors. So if we zoom in and magnify part of it, okay, you can see that each little square that you can see is a separate square packed together to make up this picture, and then different colors of tile or stone are used to make the features of the picture. So if we look here, for instance, we're mostly pale grey stone and then a few different coloured tiles inside it. Okay, so this is the picture of the cell membrane. You can see that most of these lipid heads are rather like all these little stones, and I think that this is what gave the idea of a mosaic. It's a mosaic of little pieces put together, and then every now and again you get something that's different, that's floating in that mosaic. Okay, and then sometimes there's something that's very different, like these proteins, and they are also part of the same picture, but they're different from what makes up most of the picture. Okay, so if most of this picture is grey tile, some of it is black tile or yellow tile. Okay, do you see what I mean? And this technique, this way of making a picture, is a mosaic. So that is why this is called the fluid mosaic model. So just to remind us, proteins are like icebergs floating in a lipid sea. Okay, so let's think about those proteins. What are some of the functions of membrane proteins? Well, this diagram just summarizes some of the things they can do. They can be transporters, allowing things to be transported from one side of the membrane to the other. They can be receptors, receiving a signal on one side of a membrane and passing that signal to the other side of a membrane. They can be enzymes, which are anchored in the membrane to keep them in place, but which are carrying out a catalytic reaction, an enzymatic reaction, and changing another molecule. Or they can be anchors holding that membrane in relation to other parts of the cell, for instance, the cytoskeleton or the nuclear lamina. Okay, so functions of membrane proteins. So if we think about how a membrane protein is able to be associated with a membrane, I hope that you've taken notice of the lectures you've already had about proteins and about amino acids and about the way that different amino acids have and therefore can give proteins different hydrophobic and hydrophilic characteristics. So we've said that the inside of the lipid bilayer of the membrane is hydrophobic. The fatty acid tails of the phospholipids make the inside of a membrane a hydrophobic, a water-free environment. So a protein that has a hydrophobic part can very easily associate with the middle of the cell of the membrane structure, the lipid bilayer, and it would be difficult for a protein like this to be away from the membrane, because then this hydrophobic part of the protein would be exposed to water. It would be energetically unstable. So here is an alpha helix inside this protein, which is made up of hydrophobic amino acids, which allows this part of the protein to be embedded right through the lipid bilayer of the membrane. And then there is a hydrophilic part of the protein on both sides of the membrane that's able to associate with a watery environment on the inside and the outside of the cell. Okay, so there are a number of different ways that membrane proteins can have this sort of hydrophobic part. This is what we've already seen. This is a very similar protein to this one with a hydrophobic alpha helix in the middle and a hydrophilic head and tail. But this particular protein also has a lipid anchor. Part of the protein is actually a lipid, and that also makes an association inside the membrane. And we see the same thing here and here. This protein, number 2, has several alpha helices. So it has a hydrophilic part, a hydrophobic helix, hydrophilic part loop, a hydrophobic helix, a hydrophilic loop, a hydrophobic helix, and another hydrophilic part. Okay. Number 3 here is what's called a beta barrel. So this is a number of sections of beta sheet lying side by side and rolled up to make a circle. So this is quite a common structure for membrane proteins. So the beta sheet would be made up of hydrophobic amino acids, and they roll up in a way that leaves a hole in the middle. Okay, and we'll be talking about that sort of thing later. Number 4 here is a peripheral protein that's anchored in the lipid by an alpha helix of hydrophilic amino acids, but this one's anchored just on one side of the membrane. Okay, so this one, it has parts on both sides of the membrane. This is just on the inside of the membrane. This is somehow similar, but rather than having a hydrophobic alpha helix, it has a lipid anchor, which is holding the membrane, the protein onto the membrane. And this one is similar. This has an anchor which is holding it on the outside of the membrane. These two last ones are proteins which are somehow similar to this one with an alpha helix and two hydrophilic parts. But in this case, the hydrophilic part on the inside of this one and on the outside of the other one is forming non-covalent interactions with another protein and therefore holding this second protein close to the membrane, even though the second protein isn't actually anchored into the membrane itself. So a number of different ways that membrane proteins can stay associated with the membrane, but all to do with hydrophobic and hydrophilic interactions. Okay, a pause for questions if you have any. Okay, so let's move on now to think of some of the properties of the membrane of a phospholipid bilayer that come from this structure. So the first one I've already mentioned, the fluid mosaic model, is called the fluid mosaic model because the molecules involved in it are fluid. They can move around. So each of these lipid molecules in the bilayer is not fixed in place. It can move around quite easily in the plane of this bilayer. So it's quite difficult for a lipid to move from this side of the bilayer to this side, but it's very easy for it to move around within the layer that it's part of. And this is how the bilayer can be a lake that other molecules can float in it. So why is this important? Well, different membranes can have different degrees of fluidity. They can be more or less fluid, and how fluid they are depends on the interactions between the molecules, most particularly between the interactions between the fatty acid tails of the lipid molecules. It's also very dependent on temperature. So if the temperature is reduced, if a membrane structure is cooled down, there is less movement in the lipid bilayer. The fluidity decreases. If the temperature is increased, there is more movement and a lot of movement around. And the membrane, if it's increased enough, the membrane becomes less stable and breaks down. Okay, so if we think about the sort of shapes that phospholipids can make, this all depends on the particular shape of the phospholipid molecule. So here we see a lipid that has a hydrophobic head and just a single tail. Most of the lipids we've looked at so far have had two tails like this. This one just has two. So its head is wider than its tail. It's a wedge-shaped molecule. And if a large number of molecules of this shape interact together, they form a tiny sphere like this, okay, with hydrophobic tails in the middle and hydrophilic heads on the outside. And this is called a micelle. On the other hand, we've seen that phospholipid molecules found in membranes tend to have two tails like this, which means that their tail and their head are a similar diameter. They're more like a cylinder rather than a wedge. Okay? So when molecules like this associate together, rather than forming a spherical structure like this, they form a flat bilayer. Okay? So this bilayer is exactly what we need for a cell membrane. So the aggregation, the formation of a correct phospholipid bilayer structure for a cell membrane depends on the nature, on the shape of the phospholipid molecules that make it up. If we move on to look at this structure, which is called a vesicle, here again we've got the cylindrical phospholipid molecules, but we've got more of them on the outside than on the inside. Do you see? So we've got a smaller surface on the inside than on the outside. And this means that there is an aqueous cavity, a watery hole, a water-filled compartment trapped inside this bilayer sphere. Okay, and I hope that you can see this is the start of the sort of cell membrane that we're talking about. If we think about the outer membrane of a cell, we have the bilayer outside the cell and all the contents inside. If we think of a mitochondrion, we have the lipid bilayer around the outside and the contents of a mitochondrion on the inside. If we think of a lipid transport vesicle, we have the rest of the cell on the outside and the contents of the vesicle on the inside. So this is the sort of assembly that's very important in forming the structure we see in cells. If we take a solution of phospholipids and just shake them up with water, we see them forming this sort of vesicle just by themselves. It's a very stable structure. It's energetically stable in relation to lipids interacting with water. It doesn't require energy or enzymes or anything to build this sort of structure. It comes about just because of the nature of the phospholipid molecules themselves. And I talked last time about the way the nuclear membrane of a cell can break up into little vesicles when it dissolves, when it breaks down during mitosis. Well, this is the sort of vesicle it forms, but with its proteins, with its nuclear pores and other proteins, also embedded still in these vesicles. So very quickly, when it's needed again, these vesicles can come together, and just by touching together, they can open out and form a larger bilayer sheet. Okay. So another important property of the phospholipid bilayer is its permeability. What molecules are able to pass through that lipid bilayer? So here is a lipid bilayer, just indicated in gray here. And here are different groups of molecules and how easily they can pass through this membrane. So hydrophobic molecules, molecules that are able to dissolve in the hydrophobic inside of the lipid bilayer, can pass through very easily. Okay, so they are things like oxygen, carbon dioxide, nitrogen, and steroid hormones. They are able to dissolve in the lipid and they can just diffuse freely from one side of the membrane to the other. But remember, because these molecules are hydrophobic, they don't dissolve very well in the watery part of the cell, so they can pass easily through the membrane, but there isn't very much of them dissolved in the aqueous part of the cell. uncharged polar molecules like water itself or urea or glycerol are able to pass to a small extent through the membrane, but a much lesser extent than the hydrophobic molecules. Okay, some pass through, most are not able to pass through. Larger uncharged polar molecules, so these are small ones, these are larger ones, and the examples here are the sugars, glucose and sucrose. A smaller still amount can pass through, but most can't. And then charged molecules, which have a very good interaction with water, but are very hydrophobic because of being charged. Things like hydrogen ions, sodium ions, potassium ions, and so on. They cannot pass through the lipid part of a membrane. Okay, so how permeable the lipid membrane is, or the lipid part of the plasma membrane, depends on what molecules are trying to pass through it. Okay, any questions? Okay, so I've just shown you that many important molecules like sugars, for instance, glucose and sucrose, or ions like calcium or sodium or potassium or chloride, are not able to pass through the lipid of the membrane. So if you're wondering, how can these molecules get into the cell if they can't get through the membrane? Well, the answer is that the cell is able to transport them through its membrane using the other molecules we've seen that are embedded in that plasma membrane. And this allows the cell to be selective, to choose, to select which molecules are allowed to enter and leave the cell. So we can describe the cell membrane as a whole as being selectively permeable. It is permeable, things can pass through it, but selectively permeable. The cell selects what it allows to pass through it. And very important in this process are those membrane proteins. Okay, so proteins, we've already seen, have a number of different functions. They can be involved in transport across the membrane. And if we look at this diagram we've seen before, notice that this protein has a hole through the middle. So we're looking at a sort of cross section. It's been cut through the middle so we can see its hole. And this is a transport protein or a protein channel. This is a protein whose function is to allow transport across the lipid bilayer of the cell membrane. Okay. So we've already talked about the way that proteins can be embedded in the membrane. And different functions. So this is what we're thinking about now, the transporter, how a protein can transport molecules from one side of the membrane to the other. So here, this is the inside of a cell and this is the outside. So these molecules are being transported out of the cell. Okay, so we've already mentioned diffusion. I hope you understand what diffusion is. If you have forgotten what you've already learned about diffusion, please do make sure you understand. Diffusion is the movement of molecules from a high concentration to a low concentration. So some molecules can diffuse through the cell membrane. They are the hydrophobic molecules that we've seen. There are also two types of transport that can move other molecules through the membrane: passive transport and active transport. And the difference between passive and active transport is whether it needs metabolic energy or not. So passive transport does not need metabolic energy. Active transport does need metabolic energy. So passive transport is sometimes called facilitated diffusion because, just like diffusion, it moves molecules from an area of high concentration to an area of low concentration. So here, if we look at these little yellow molecules, there's a lot of them on this side of the membrane and not so many on this side. And the way they're able to pass through this membrane is through a channel protein that has an aqueous pore in the center, a watery hole. So anything that's dissolved in the water on one side of the membrane and is small enough to physically fit through the narrowest part of the channel is able to diffuse across the membrane from high concentration to low concentration. So it's made easier because of these proteins, but it is still basically diffusion. Okay, so this is a way that hydrophilic small molecules can pass through the membrane. They stay dissolved in water. They don't have to go through the hydrophobic lipid part of the membrane. They stay dissolved in water, but can pass from one side of the membrane to the other. A second way of passing through the membrane is to use a transporter protein. So this is slightly more complicated. This is a protein, again, it's embedded in the membrane, but this is an allosteric protein. Remember what that means. It's a protein that changes its shape when it binds to a ligand. So here, the protein has a binding site for its particular ligand, for a particular solute, which needs to pass through the membrane. And when that ligand binds to the protein changes shape so that the solute can be released on the other side of the membrane. Okay, so there's a conformational change, a shape change happening and allowing the small molecule to pass through the membrane. So here we're just diffusing from high to lower concentration. Here we're also moving from high to low concentration, but by way of a transporter molecule that changes its shape. Okay, so both these sorts of passive transport move the solute molecules from an area of high concentration to an area of low concentration. It's exactly the same as simple diffusion, just making use of the transporter molecules to solve the problem of hydrophilic molecules getting through the hydrophobic cell membrane. Okay, so I want to talk about the special case of water molecules, the passive transport of water molecules, or osmosis. So this is somehow a special case of facilitated diffusion. So we've already seen that this sort of channel protein with an aqueous pore in the middle that molecules can pass through. Now imagine that these little yellow molecules are the water molecules themselves. So the channel protein allows the water to move from one side of a membrane to the other side. And the movement of water molecules across the membrane depends on the concentration of solute molecules, molecules that are dissolved in the water on both sides of the membrane. So I think that you will have learned at A level that osmosis means the movement of water molecules from an area of an area of low solute concentration to an area of high solute concentration. Okay? And you might have become confused. I know that when I first learnt about this at school, I became very confused. But diffusion goes from high to low, but osmosis goes from low to high. But think it through. If there is a lot of, let's say, salt dissolved in water, a high concentration of solute, there is less water there. Okay? If there is a low concentration of salt, there is more water there. So if we think of the water molecules that are moving, they are actually still moving from a high concentration of water molecules to a low concentration of water molecules. So that is what's happening in osmosis. Water moving from a high concentration of water molecules, which is a low concentration of solute molecules, to another place where there is a low concentration of water molecules, therefore a high concentration of solute molecules. The special membrane channels through cell membranes which allow water to pass are called aquaporins, water pores. Okay, aquaporins. Okay, so some more words that are associated with osmosis. So if we compare two solutions separated by a membrane, if I start here with an isotonic solution, so here we have equal concentrations of solute on both sides of a membrane. So if we imagine that this is a cell, this is the inside of a cell, and this is the outside, the solute concentrations are equal, so there is no net movement of water. There might be some movement across this membrane, but it's equal in both directions. Okay? If we think of a hypertonic solution, where there are more solute molecules outside the cell than inside, in this case, water will move out of the cell and the cell will shrink up. And this would happen, for instance, if you took a plant and put it in salty water, you would see the plant become limp as the water moves out of its structure, out of its leaves and stem. On the other hand, hypotonic is where there is more solute molecules inside the cell, and therefore water moves into the cell. The cell becomes turgid. The water molecules push against the outside membrane of the cell. The cell becomes very stiff, very turgid. Okay. So the concentration of the solutes inside and outside the cell are what determines what makes the movement of water across the membranes. So cells are able to control how much water is inside them by controlling the concentration of solutes inside them. And this is especially true of small ions like sodium and calcium and potassium ions. Okay, pause for questions. Okay, so another sort of channel we find through the cell membrane is an ion channel. So these are channels which are similar to the porin channels we've seen already, but these are able to open or close. Okay, so again, we've got an aqueous pore in the center and a channel that allows passage from one side to the other. So again, this is passive transport, that the ions are diffusing down their concentration gradient. But the channels, the channel proteins are another allosteric protein. They're able to change their shape when they receive a signal, and they can change between an open and a closed form. These channels can be specific for different ions, particular ions that are allowed to pass through these channels. So being able to open and close is called being a gated channel. And when the gated channel receives a stimulus, they are able to open or close appropriately and allow the correct ion to pass through them. So what sort of signal can make these gated channels open and close? Well, a very important signal is our voltage gated channels. So these channels open when the voltage across the membrane changes. And this is very important in the transmission of nerve impulses. I think that you might have already learned in physiology about what happens when a nerve impulse is sent as an electrical signal along the length of a nerve and is received at the end. There is a flood of ions which allows that signal to pass from one cell to the next. And those ions are able to flood out because the gated pores open, the gated channels open, and allow those ions to suddenly move from high to low concentration. There are also mechanically gated channels which open when there is an actual stress put on the protein. So here we're seeing things where there is a movement being produced because of the interaction between the protein opening and the passage of ions. There is also a type of gated channel which opens when a ligand binds to it. So something could be binding and telling the channel to open. So the ligand would be being received as a signal. Okay, so let's have a look at some things that can be transported by transporter proteins. Here is just a cell illustrating different sorts of transport. So here we're seeing ion transporters that we've already talked about. So here we're seeing some red ions being specifically transported into the cell, and some blue ions transported out of the cell. So these could be something like sodium and chlorine ions being transported in different directions, or potassium and chlorine ions. Here we see some amino acids being transported into the cell. So here the picture is that the amino acids are needed as protein building blocks inside the cell. So they're being specifically transported into the cell so that they can be used. And in the same way here, we're seeing a glucose transporter. Glucose, a sugar and energy source that's needed inside the cell, so it's being transported across the membrane. But all of these are examples of passive transport, but they only happen from high to low concentration. Okay, so now let's think about active transport. So this is the type of transport that it needs energy. And the reason that it needs energy is that active transport allows molecules to move against their concentration gradient. So rather from moving from high to low concentration, active transport can move molecules from a low concentration to a high concentration, which is an energetically less stable situation. So energy needs to be used to actually move those molecules. So here we see a number of different ways that energy can be used in transport. Here is a very common method. It's an ATP driven pump. So the energy from ATP, the energy currency of the cell, is being used to pump a molecule against its concentration gradient. Here it is a similar molecule, but this is getting its energy from light. So this is the sort of situation that we see during photosynthesis, where energy from light is used to move molecules against their concentration gradient. Here we're just indicating the gradient. The third method we see here is coupled transport. So here we've actually got two types of molecule that's moving across the membrane. The yellow molecule is moving against its concentration gradient, but the red molecule is moving with its concentration gradient. Okay, so it's a high concentration here, low concentration here. And by moving from high to low, this coupled transporter is able to also move the yellow molecule from low to high at the same time. Okay, so active transport can get the energy in several different ways, but they are all forms of active transport. I think that you'll already be familiar with the sodium potassium pump. I think that you will have looked at this in physiology. Very important in receiving signals, nerve signals, as sodium and potassium are pumped across a membrane. So this is an example of transport that's powered by ATP. And it pumps sodium out of a cell against its concentration gradient. So here is the outside of the cell, here is the inside. The sodium is at a higher concentration outside the cell, but it's still being moved against its gradient. And potassium is being moved into the cell. So the potassium has a low concentration outside and a high concentration inside. So it is also moving against its gradient. So it's the energy from the ATP that allows this pump molecule to move the ions against their gradients. So each ATP molecule is able to move 2 potassium ions into the cell and 3 sodium ions out of the cell. So if you follow through the diagram here, the sodium ions are bound onto the outside of a transporter and ATP is used. This energy allows the transporter to open and to take the sodium ions into it, and this change in shape opens binding sites on the outside for the potassium ions to bind. So now the sodium ions are released on the other side of the membrane. The potassium ions are bound and allowed to enter, pass through the membrane, and be released on the inside of the cell, resetting the molecule to its original condition so that the sodium is able to bind again. Okay, so it's a pumping motion, a cycle that's repeated using ATP, breaking ATP down to ADP, an inorganic phosphate each time, and transporting 3 sodium ions out of the cell and 2 potassium ions into the cell. Okay, any questions? Okay, so this just summarizes the different sorts of transport that we've seen so far. So we've seen diffusion across the lipid bilayer, and we've seen facilitated diffusion, channel mediated diffusion, and transporter mediated transfusion, but these are all forms of passive transport which don't need energy. And then we've seen active transport, which needs energy and is able to move molecules against their concentration gradient. Now we're going to talk about other forms of movement, exocytosis and endocytosis. Okay, so the question we're asking here is how can we transport very large molecules from one side of a cell membrane to the other, from outside to inside or inside to outside? So examples of the sort of molecules we might want to transport are polypeptides or polysaccharides or polynucleotides. These are too large to be transported by the protein carriers, and they're too large to diffuse through the cell membrane itself. So these are transported across the membrane by using vesicles. So we saw before what the structure of a vesicle looks like. It's a sphere of membrane bilayer with an aqueous center. Okay, so here is a vesicle. We have a membrane bilayer, and this circular structure is a sphere that's cut across the middle so we can see what's inside. So we've got a watery center with things dissolved in it, wrapped up, enclosed in the phospholipid bilayer of a membrane. And if we looked at the cell surface here to see how that can happen, we can see that the plasma membrane, because it's very fluid, because it can change its shape, is able to pull in and make indentations. Okay, and these indentations can then be closed off and broken off and become separate vesicles. And when they do this, any liquid that's inside the pinched off bit of membrane will be inside the vesicle. So this is the process called pinocytosis. Here we see a similar thing happening with an even larger molecule, a food particle, perhaps a lot of molecules here. And this is phagocytosis. And here is receptor-mediated endocytosis, which we'll talk about in a minute. But the common feature here is that all these things that end up inside the vesicles, inside the cell, are too large to pass through the cell membrane by the transport mechanisms that we've seen so far. So the first thing we're going to talk about is endocytosis. So endo means inside, cytosis to do with a cell. So we are bringing things inside the cell. The plasma membrane forms a vesicle around the substance which needs to be taken into the cell. Okay, so the first type of endocytosis is phagocytosis. So this is just a word which means cell eating. Phago, a bit like esophagus, okay, it's to do with eating. And here is an electron micrograph of a macrophage, a cell of the immune system, eating a worn out red blood cell, okay? So you can see that the whole red blood cell is being taken inside the macrophage cell. So the way the macrophage is doing this is to put out pseudopods, that means false feet, okay, on all around the cell it's engulfing, or the particle it's engulfing, until the membrane meets on the other side. Okay, so here is the solid particle, here is the cell membrane being pushed out in pseudopods, joining up on the other side and breaking off to form a food vacuole or a vesicle inside the cell, where it can be transported in the cell and dealt with. It can be digested in this case, for instance. So if you look carefully at what's happening here, you'll realize that what is the outside of the cell membrane here becomes the inside of a vesicle, and what is the inside of the cell membrane becomes the outside of the vesicle. Okay. Pinocytosis is very similar, except the molecules involved are too small to see in a microscope. For phagocytosis, people looking through microscopes could see the particle that was being taken in. They're large enough particles to see. For pinocytosis, they could see the cell doing this sort of indentation and taking things in and forming vesicles, but they couldn't actually see what was being taken into the cell. So they gave this the name of pinocytosis, which means cell drinking. So small molecules or liquids, water, are being taken into the cell by pinocytosis, but exactly the same pattern, pinching off of a little part of the membrane and a vesicle being formed. The third thing I want to tell you about is receptor-mediated endocytosis. So here, the same thing is happening, but the cell is being very selective about what it brings into the cell. So here we have receptors on the outside of the cell, which are detecting the presence outside the cell of a particular molecule, shown as these yellow stars, a molecule that the cell wants. So this might be a food source or a particular nutrient that it needs, or something that's needed inside the cell. The receptor finds it there on the outside of the cell, and it acts as a signal to start this process of bringing it into the cell. So again, we're making a vesicle, but notice this time that on the inside of the membrane, where there are receptor molecules on the outside, there are coat proteins on the inside. And the structure that's being formed here is called a coated pit. And because of the way the vesicles form, when we form a vesicle, we have the receptors with their ligand bound on the inside of the vesicle, and the coat proteins are on the outside of the vesicle. So in this case, this is almost like a little container with a label on it. So if you imagine going into the shop and buying some food in a can, you only know what's inside that can because of the label on the can. Okay, so these coat proteins act like a label. It tells the cell what is on the inside of the vesicle by the particular coat proteins that are on the outside of the vesicle. Here is an electron micrograph of the same process happening. So I think you can see here that a pit is forming. This is a coated pit, and along the inside of this pit, there are these coat proteins. On the outside, there are the receptors binding to particular ligands. And because the ligands are present, the pit deepens and pinches off and forms a vesicle. And now the vesicle has the ligand attached to the receptors on the inside and the coat proteins on the outside. So the type of protein that's on the outside is often called a clathrin. Okay, so type of protein that labels these coated vesicles is called clathrin. Okay, so if vesicles are containing things from outside the cell, or brought into the cell, what then happens to them? Well, often they fuse together to form an endosome. That means a body, a structure inside the cell. And then the endosome can fuse with a lysosome, and the contents, the food and the other molecules from outside the cell that have been brought inside, can be digested, broken down into their building blocks, and then the building blocks, the amino acids or the carbohydrates or whatever, can be used by the cell. So the opposite process to endocytosis is exocytosis. And here we've got exactly the same thing happening, but in the opposite direction. So here we have a secretory vesicle. So this is a vesicle containing something that's been made by the cell, but needs to be released outside the cell. So this could be a number of different things, but a common example would be a hormone that's been made in the cell and needs to be released outside the cell into the bloodstream where it can act as a signal to other cells. So the membrane enclosed vesicle moves until it makes contact with the plasma membrane of the cell. And because of the nature of the lipid, because of how fluid it is, it's very easy for that vesicle of lipid to fuse with the membrane of the cell. The lipid molecules move and become part of the bilayer of the cell itself. And that allows the contents to be released outside the cell. Okay, so here it's moving towards the membrane, making contact with the membrane, fused with the membrane, and then opening out and releasing its contents on the outside. So you'll realize that as it does this, it's adding more molecules to the number of molecules in the cell membrane. So if a lot of vesicles were moving and making contact with the cell membrane at the same time, the cell membrane would actually expand, it would get larger. So there has to be a balance between endocytosis and exocytosis, vesicles being pinched off and brought into the cell, and vesicles fusing with the cell membrane and releasing contents outside, but overall the size of the cell membrane stays the same. Okay, so here's just an electron micrograph of insulin being secreted from pancreatic cells. So here is a vesicle that has just reached the cell membrane with its contents of insulin. And here is a second vesicle, which is already fused with the membrane. And here is the insulin protein ready to be released outside. Okay. So I'm just going to show you another video now, which just animates a lot of the things we've been talking about. And I hope this will help you to visualize what's going on and to fix what we've been talking about into a big picture, if you like.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, Both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
Okay, I hope you enjoyed that. I think it just helps you to visualize these different sorts of transport and what's going on. Okay, I thought I'd finish with just a few words about why it's important for you as pharmaceutical scientists to know about transport across membranes. There are a number of very relevant questions that you need to ask if you're thinking about making drugs. So most drugs need to act inside a cell. To get inside the cell, they must cross the cell membrane. So how is the particular drug that you're interested in going to get inside a cell? Can it be taken in by endocytosis? Can it be taken in by transport? Will it diffuse into the cell? These are questions that need to be answered about any particular drug. Another important thing is that membrane transporters are so important for so many of our metabolic processes or physiological processes that they are often themselves the target for drugs. So increasing or decreasing transport across membranes is often the aim of drug treatment, to affect the cellular processes by changing that transport. So for instance, high blood pressure or hypertension uses drugs which decrease calcium transport across membranes. The third thing I thought I'd mention is drug resistance, antibiotic resistance. This is often caused by the cells, the bacterial cells that the antibiotic is aiming to kill. The cells are able to become resistant to that antibiotic by developing transporters which remove that drug from the cell, which pump it out, which stop it having its effect on the cell. So thinking about transporters and transport across membranes is very important when you think about how a drug works. Okay, I'm going to finish there for today. We'll continue to think about membranes next time, when we'll look at the nuclear membrane in a bit more detail, and then we'll go on to think about interactions between cells. But just to send you to some textbooks, the material I've covered today comes from chapters 10, 11, 12, and 13 in Molecular Biology of the Cell, but it's not the whole of the chapters. There's a lot in those chapters which I haven't covered. It's all good, it's all interesting, but it's such a big topic that I can't cover all of it. But that's where you'll find what I've talked about today, and you might want to read more deeply and read some of the rest of those chapters as well. If you want to go to Leninger, chapter 10 is about lipids, and you'll be coming back to that when you look at lipids in the main biochemistry lectures, and chapter 11 is about membranes. Okay, so I'll finish there, and another video for the next lecture in a few days' time.